In this video, we're gonna study about control of translation in eukaryotes and differences between prokaryotic and eukaryotic translation. So we all know what is translation. Translation is a process where mRNA is converted into protein. Then what is control of translation? So translational control is a process where it controls mature mRNA to be translated or not. And also, it controls how long a mature mRNA is able to remain in the cytoplasm for translation. It plays an important role in modulating the expression of many genes that respond to endogenous or exogenous signals, such as nutrient supply, nutrient stress, or nutrient hormones. It plays an essential role in regulating gene expression both in prokaryotes and eukaryotes. So, we can see translational control in both prokaryotes and eukaryotes, but because of the different natures present in both prokaryotic and eukaryotic mRNA, the translational control varies. Like uh, polycystonic mRNA is in prokaryotes and monocystonic mRNA is seen in prokaryotes. And also the absence of nuclear membrane in prokaryotes forms a different possibility for transitional control. Whereas in eukaryotes, the structure formed in the regions of mRNA can be uncertain for ribosome binding site, thus reducing translation of some cystons relative to others. The stems and loops formation in mRNA ribosomes can inhibit endonuclease and give certain regions of polycystonic mRNA, thus having a great half-life than others. The greater half-life of mRNA causes greater stability, thus causing more translation and more proteins are synthesized. An interesting form of transitional control is seen when several operons encoding ribosomal proteins, like in E. coli, where the mRNA has a tertiary structure that resembles the binding site for ribosomal protein, which is encoded in mRNA. When there is insufficient rRNA available for translation, the product to bind to will bind to its own mRNA, which which prevents further translation. In prokaryotes, sometimes it forms duplex near the ribosome binding site of certain mRNAs due to short antisense RNA molecules, thus inhibiting translation. Eukaryotes generally control the amount of specific proteins by, by the level of transcription varying, varying the level of transcription of the gene by RNA processing. But some controls occur in cytoplasm, where you can see the translation occurs in eukaryotes, both in nucleus and cytoplasm. The presence of multiple copies of 5' AUUA to 3' in eukaryotes is usually in a 3' non-coding region that marks the mRNA for rapid degradation, which causes the limited translation due to mRNA degradation, which stops translation. And another form of transitional control that involves protein binding directly to the mRNA. This prevents translation. And this type of translation where the mRNA is called masked mRNA. In appropriate circumstances, the mRNA can be translated when the protein dissociates. In cytoplasm, some non-coding sequence can cause mRNA to be located in specific part. When it is translated, this gives rise to a gradient of protein concentration across the cell. And another interesting and specific use of translational control in eukaryotes is that it regulates the amount of iron in the cell. The iron is required for the essential activity of some proteins, but it is harmful in excess. The iron is transported into cells by a protein receptor called transferrin, and this is stored within cells bound to a storage protein called ferritin. In eukaryotes, the mRNA contains non-coding sequence for each of the proteins that can form stem loop structures called the iron response protein. So the iron response protein is involved in control of iron metabolism by binding mRNA to repress translation or degrade mRNA, to which the iron response protein, the iron response element to which a 90 kilodalton iron sensing protein can bind. But however, the position of the iron response element and its action to bind with iron sensing protein is very different comparatively. In the receptor protein transfer, the mRNA, where the iron response element is in the 3' non-coding region, and when it binds to iron sensing protein, which it does when the iron is scarce, this stabilizes the mRNA and allows more translation when when iron response element is binded with iron sensing protein. But when the iron levels are high, the iron sensing protein dissociates from the iron response element. This unmasks the destabilizing sequences, which in turn which is then attacked by the nucleus. Nucleus is enzyme, thus reducing translation due to mRNA degradation. When the enzyme nucleases attacks, the mRNA is degraded, thus reducing translation. But when the iron levels are high, the transferrin receptor protein in mRNA is destroyed, but the translation of ferritin storage protein mRNA is increased when the iron levels are high. This occurs only because at low iron levels, the iron response element, which is located in 5' non-coding region, will bind to the iron sensing protein, which in turn reduces the ribosome's ability to translate into ferritin mRNA. But when the iron level rises, the iron sensing protein, it will again dissociate from iron response element. But in this case, we can see there is an increase in translation of ferritin. This is because of the ribosome's progress is not hindered when the iron levels rise. This translation control system rapidly and responsively regulates the intracellular iron levels. And we can see another example for translational control is that the protein binding in the vinicity can of destabilizing sequences and inhibiting them in translational control. And another example of translation control, which includes when microRNAs in eukaryotes, which binds to the mRNAs, which are complementary to each other, they cause either degradation or translational repression of mRNA, where elongation initiator factor 4E is the inhibitory protein that prevents the cap dependent initiation of translation, which causes translation repression or translation, which causes translation repression or mRNA degradation. In eukaryotes, the small molecules, for example, like thymine, which will bind to shine Dalgorno sequence, that is 5' AGGA GG to 3' is a shine Dalgorno sequence, which is only found in prokaryotic mRNA that prevents ribosome binding. So you can see the ferritin translation regulation, where the iron response protein will bind to the iron response element when the iron levels are low in the absence of iron, this causing inhibition of ribosome, where it binds to iron response protein in 5' UTR. This prevents the translation of mRNA. Thus, there is no production of ferritin. So even here you can see when in 5' to 3' mRNA, when iron element is low, a translational repressor will bind to the ferritin mRNA. This causes where translation is blocked and there is no ferritin is produced. Now let us study the difference between prokaryotic and eukaryotic translation. So the prokaryotic and eukaryotic translation, both are involved in protein synthesis by decoding the genetic instructions carried by the mRNAs for producing proteins. This prokaryotic and eukaryotic translation contains codons, that is nucleotide triplets on mRNA, and are translated into sequence of amino acid. Both prokaryotic and eukaryotic translation share a same basic plan throughout the process. The main difference between prokaryotic and eukaryotic translation is that the prokaryotic translation occurs synchronizingly with its transcription, whereas eukaryotic translation occurs asynchronizingly with its transcription. So now let us study the difference between prokaryotic and eukaryotic translation in terms of timing. So in prokaryotic translation, the both transcription and translation are simultaneous process because it takes place only in the cytoplasm, whereas in eukaryotic translation, it is a discontinuous process where eukaryotic translation takes place in nucleus as well as in the cytoplasm of the cell. When it comes to ribosome, prokaryotic translation contains 70S ribosome, where 30S ribosome is smaller subunit and 50S is the largest subunit. In eukaryotic translation, it contains 80S ribosome, where 40S is a smaller subunit and 60S is a larger subunit. In, in terms of ribosome's messenger RNA source, the prokaryotic translation contains mRNA in the cytoplasm and it is polycystonic. The prokaryotic mRNA occurs in the cytoplasm, whereas in eukaryotic, the mRNA occurs in nucleus. After post-transcriptional modification, they are released into cytoplasm by nuclear pores. And in eukaryotic translation, the mRNA is monocystonic. Monocystonic. When it comes to lifespan of mRNA, the prokaryotic translations mRNA are very unstable and this lives for few seconds to 2 minutes. But in eukaryotic translation, the RNA are quite stable because they are monocystonic and they live for few days to few hours. The difference in sequence in the untranslated region in prokaryotic translation, we shine Dalgarno sequence is found in the 5' UTR, which has 10 nucleotides upstream to the start codon, where in eukaryotic translation, we can see Kozak sequence is found, that is 5' UTR, a few nucleotides upstream to the start codon. The difference in location is in prokaryotic, the translation occurs in cytoplasm and it is performed by 70S ribosome. Whereas in eukaryotic translation, it is performed in the endoplasmic reticulum, which is attached to the nucleus. And here the ribosome is 70S. The localization in cell cycle. The difference in prokaryotic and eukaryotic translation in localization in the cell cycle is that the prokaryotic translation has no definite phase for their occurrence, but in eukaryotic translation, this occurs in G1 and G2 phases of the cell cycle. The difference in translation initiation is in prokaryotic translation, it is a cap independent initiation. But in eukaryotic translation, it is both cap dependent and cap independent. When the cap is present, the translation occurs, but when the cap is in the absence of cap, the mRNA degrades, thus no translation occurs. The difference in initiation factors in terms of prokaryotic translation, there are 3 initiation factors that is involved in translation, that is initiation factor 1, initiation factor 2, and initiation factor 3. But in terms of eukaryotic translation, there are totally 9 initiation factors that are involved, that is elongate eukaryotic initiation factor 1, 2, 3, 4A, 4B, 4C, 4D, 5, and 6. The difference in first amino acid is in prokaryotic translation, the N-formyl methionine is the first amino acid which is added to the polypeptide chain. But in eukaryotic translation, methionine is the first amino acid added to the polypeptide chain. When the difference in elongation factor is that in prokaryotic translation, there is 2 elongation factors that are involved, that is elongation factor G and elongation factor TU. But in eukaryotic translation, even here it is 2 elongation factors that is involved, that is eukaryotic elongation factor 1 and eukaryotic elongation factor 2. In terms of speed, Prokaryotic translation is a faster process and which adds up to 20 amino acid per second. But in eukaryotic translation, it is a slower process which adds only a single amino acid per second. The fate of first amino acid. In prokaryotic translation, the formal group, that is N-formyl methionine, is removed from the first amino acid, retaining methionine in the polypeptide chain. Only the formal group is removed and the methionine is retained from the fate of first amino acid. But in terms of eukaryotic translation, the whole methionine is removed from the polypeptide chain during translation. The difference in release factor are in prokaryotic translation, two release factors are involved. That is release factor 1 from UAG and UAA codon and release factor 2 from UAA and UGA codon. In terms of eukaryotic translation, a single release factor is involved, that is eukaryotic release factor 1. So now let us summarize the difference between prokaryotic translation versus eukaryotic translation. So in prokaryotic translation, we can see it is a simultaneous process with its transcription and translation. But in eukaryotes, it is a discontinuous process between transcription and its translation. In prokaryotic, there is presence of 70S ribosome in which 30S is a smaller subunit and 50S is a larger subunit. But in eukaryotics, we can see 80S ribosome where 40S is a smaller subunit and 60S is a larger subunit. In prokaryotic mRNAs, it occurs in cytoplasm because it is a prokaryotic cell, and eukaryotic mRNAs, it occurs in nucleus. Later, it moves through nucleus pore into the cytoplasm. Wherein prokaryotic, the mRNA are very unstable because they are polycystonic and they live for few seconds to minutes. But in eukaryotes, the mRNA are quite stable and live for few hours to days because it is the eukaryotic mRNA are monocystonic. And in prokaryotic, it is the translation is performed by 70S ribosome in the cytoplasm, whereas in eukaryotic, the translation is performed by the 80S ribosome, which attaches with the endoplasmic reticulum. In prokaryotics, there is no definite phase for the occurrence, but in eukaryotics, it occurs in G1 and G2 phases in the cell cycle. Prokaryotic translation is a cap-independent initiation, whereas in eukaryotic, it is a cap-dependent and cap-independent initiation. In prokaryotic translation, there are three initiation factors which are involved in initiation process. That is initiation factor 1, initiation factor 2, initiation factor 3. And there are 9 initiation factors involved in initiation process of eukaryotic translation, that is eukaryotic initiation factor 1, 2, 3, 4A, 4B, 4C, 4D, 5 and 6. Prokaryotic translation is a faster process when it is compared to eukaryotic. Eukaryotic is a smaller process, it's a slower process. A single release factor is involved in terms of prokaryote, that is eukaryotic release factor 1 and 2 release factors are involved in eukaryotic translation, that is RF1 and RF2. So let us conclude both prokaryotic and eukaryotic translation. So the translation, it is a universal process for synthesizing proteins. As a second step in gene expression, it is seen in both prokaryotes and eukaryotes. There is presence of all 20 amino acids, which is shared by both prokaryotic and eukaryotic in translation process. This both process of translation of both eukaryotic and prokaryotic occurs in cytoplasm, complete, completing the four process, that is initiation, elongation, translocation, and termination. So we can see the main difference between prokaryotic and eukaryotic translation is that the prokaryotic is a simultaneous process with its transcription, whereas the eukaryotic translation is a discontinuous process from its transcription. So these are the references, and thank you for listening.